Hello students, welcome again to EPG Pathshala. I am Dr. Chirantan Raval from Dr. APJ Abdul Kalam Government College Silvassa, that is there in UT of Dadra and Nagar Haveli. Today we will discuss about glycogenolysis, that is a breakdown of glycogen under the paper Carbohydrate Metabolism. So students, let us see what we are going to learn in this module. We will try to explore the breakdown of glycogen. We will try to understand the role of glycogen phosphorylase in glycogen breakdown. We will explore the glycogen debranching enzyme which play an important role in the debranching. Glycogen is a highly branched large polymer of glucose which is having alpha 1,4 glycosidic linkages which has branches at by alpha 1,6 glycosidic bond, which is generally present at about every 10th residues within the chain of glycogen. Glycogen is generally found in the form of granules and contains enzymes for the synthesis as well as degradation of the glycogen. Means it is found within the granules, and that granules also consists of the enzymes which is required for their synthesis and degradation. It acts as an important energy reserve. Glycogen is an energy reserve obviously for us. Suppose in a human body, if extra glucose is there, it will be stored in the form of glycogen. As and when we require, we do the breakdown of glycogen. It is stored in the liver and the skeletal muscles. Glycogen stored in the muscle will be utilized for the energy requirement of the muscles only, while the glycogen stored in the liver will be used for the energy requirement of the rest of the body. So, over here what happens? Muscular glycogen is available for the body to work only for the muscular movement, while the glycogen stored in the liver is used for the energy requirement of the rest of the body. So, in a nutshell, glycogen that is present in the muscle is available to do the task of muscles only, while the glycogen available in the liver will do the task of what you call rest of the body. We will also study the role of an important enzyme, that is the glycogen phosphorylase. Glycogen phosphorylase has a key role in this breakdown of the glycogen. Do remember this, glycogen breakdown is also known as glycogenolysis. So this glycogen phosphorylase plays an important role in glycogen breakdown. Glycogen being highly branched polymer, as we discussed, the glycogen having alpha 1,4 glycosidic linkages as well as alpha 1,6 at the branching point. So it is a highly branched polymer of glucose which needs another enzyme that helps with the glycogen debranching. So over here glycogen phosphorylase only deal with the long chain of glycogen glucose molecule. It cannot break at the point of branching. Wherever the branching occurs, that can be done with the help of another enzyme, that is called a debranching enzyme. This pathway is very important as it helps in regulating blood glucose in between the two successive meals. It means when we eat, then after the blood glucose level within the body will rise, and that's why the role of this particular enzymes are very important. When body is under starvation for a long time, we require glucose, and that glucose will be available by the process called glycogenolysis. The regulation of this both the process, glycogenesis and as well as glycogenolysis, is very important. Glycogenesis deals with the synthesis of glycogen, while glycogenolysis deals with what? The breakdown of a glycogen. So it is very important in maintaining the homeostasis of the body. These two processes, that is glycogenesis as well as glycogenolysis, are commonly regulated. There are certain hormones which stimulates the glycogenolysis like glucagon, cortisols, epinephrine, norepinephrine, while there are certain which inhibits the glycogenesis. On the other hand, insulin, which promotes the body to store glycogenesis, means insulin reduces the blood glucose. Where this glucose goes? It goes for the formation of glycogenesis. So insulin reduces the blood glucose. At the same time, glycogenesis is promoted. It is inhibiting the glycogenolysis. Glycogen is degraded by two different enzymes. First, the long chain of glucose molecule is released in muscles to fuel its contraction. It is released in the liver to transport in the blood. It is catalyzed by the glycogen phosphorylase and debranching enzyme. In the second pathway, glycogen is degraded to glucose within the lysosome by the enzyme alpha glucosidase and acid maltase. So glycogen basically degraded in two ways. One is by glycogenolysis, that involves the two enzymes glycogen phosphorylase and debranching enzyme. Another one is by lysozyme enzymes, that is specifically the acid maltase, which degrades the glycogen. The entire process is divided into three major reactions. Glucose 1-phosphate formation from the non-reducing end by glycogen 1-phosphorylase, removal of branches by the glycogen debranching enzymes, and glucose 6-phosphate formation from the glucose 1-phosphate by phosphoglucomutase. Concept map. First of all, we will study about the glycogenolysis. Glycogen is present in the cytoplasm in the form of granules. This granule contains various enzymes and regulatory proteins for both its synthesis and degradation. Glycogen is an important stored form of energy in our body. Liver and skeletal tissues are the prime location where abundant glycogen is stored. Glycogen is stored in the skeletal muscle tissue is available only for muscular activities, while liver glycogen takes up the charge of energy supply for rest of the body parts. Structure of glycogen. It is a branched polymer of glucose with alpha 1,4 glycosidic linkage that is indicated through straight, while the alpha 1,6 glycosidic bond is observed in the branching point at almost every 10 molecular units. The biological degradation of glycogen is termed as glycogenolysis. Glycogen is a highly branched large polymer of glucose molecules linked along its main line by alpha 1,4 glycosidic linkage. Branches arise by alpha 1,6 glycosidic bond at every 10th more residues. It is catalyzed by the glycogen phosphorylase and debranching enzyme. In the second pathway, glycogen is degraded to glucose within the lysosome by the enzyme alpha glucosidase and acid maltase. Glycogen metabolism is very important because it facilitates the blood glucose level to be maintained between meals and also act as an energy reserve for muscular activity. The maintenance of blood glucose is essential in order to supply energy to the tissues. This is a schematic representation of two pathways of glycogen breakdown. In one case, glycogen is broken down to glucose by the debranching enzyme and glycogen phosphorylase, while in another case, glycogen is broken down in the lysozyme to glucose molecule. Over here, alpha glucosidase and acid maltase acts. Glycogen debranching enzyme. This particular diagram explains the role of the glycogen debranching enzyme in the glycogenolysis, how it removes the glucose molecule one by one from the glycogen molecules. Glycogenolysis process. Glycogenolysis requires two main enzymes. Glycogenolysis occurs by a different pathway from glycogenesis. Glucose 1-phosphate formation from non-reducing end of glycogen by glycogen phosphorylase. That is the first step of glycogenolysis. Then removal of alpha 1,6 branches from the glycogen by the glycogen debranching enzyme. So over here the second enzyme that comes into the action. And the third one, that is the glucose 6-phosphate formation from a glucose 1-phosphate by phosphoglucomutase. This figure explains an overview of glycogenolysis. Glycogen present in the cytoplasm in the form of granules. Over here, glycogen is converted into glucose 1-phosphate with the help of glycogen phosphorylase. This glucose 1-phosphate is converted into glucose 6-phosphate by phosphoglucomutation. This glucose 6-phosphate is converted into glucose. This takes place in the liver with the help of the enzyme glucose 6-phosphatase. This glucose ultimately diffuses into the bloodstream. The glycogen stored in the skeletal muscle tissue is available only for the muscular activities, while liver glycogen takes up the charge of energy supply for the rest of the body. The entire process is subdivided into three major reactions, as we earlier discussed, that is glucose 1-phosphate formation from the non-reducing end of the glycogen, removal of alpha-1,6 branches from the glycogen, and the glucose 6-phosphate formation from the glucose 1-phosphate formation. Now, over here, we'll try to understand how glucose 1-phosphate formed from a reducing end of a glycogen by glycogen phosphorylase. Glycogen is broken down into glucose 1-phosphate by glycogen phosphorylase. It is carried out by phosphorylases reaction involving breakdown of large molecule and forming a smaller units. Inorganic phosphate is also used in this cleavage reaction. Such breakdowns of the bond by addition of the orthophosphate is referred as a phosphorylasis. A hydrolysis reaction also involves the same process, but it uses water instead of phosphate for the cleavage of bonds. Cleavage by phosphorylasis is energetically favorable because released glucose is phosphorylated. Glycogen phosphorylase act on exoglycosidic bond. Pyridoxal phosphate is a necessary cofactor in the glycogen phosphorylase reaction. This cofactor is linked to lysine that is present at the 680 residue of the enzyme. Glycogen phosphorylase will act repeatedly on non-reducing end of a glycogen chain. Glycogen phosphorylase can act continuously until it reaches 4 glucose molecule away from the alpha-1,6 branch point. Glycogen phosphorylase is an allosteric enzyme. AMP acts as an allosteric activator, while ATP, glucose 6-phosphate, and glucose act as an allosteric inhibitor. Glycogen phosphorylase is also regulated by covalent modification. For further detail, you may refer the module 26 of glycogen degradation. Generally, in the structure of glycogen, about 1 in 10 residues is branched. In such a situation, phosphorylase enzyme cannot degrade glycogen independently. It will stop or halt after the release of 6 glucose molecule per branch. This slide explains the formation of glucose 1-phosphate from a glycogen. You can see over here the role of glycogen phosphorylase. After this reaction, 1 molecule of glucose has been reduced and so the glycogen becomes N-1. Now we will discuss the second step of a glycogenolysis, that is removal of alpha-1,6 branch from the glycogen by glycogen debranching enzyme. So now let's try to understand the role of glycogen debranching enzyme. In glycogen, alpha-1,6 glycosidic bond at the branch points are not susceptible to cleavage by glycogen phosphorylase, while it can act continuously until it reaches 4 glucose away from alpha-1,6 branch point. Thus, further degradation of glycogen chain by glycogen phosphorylase occurs only after the action of glycogen debranching enzyme. Glycogen debranching enzyme shows two different activities. One is transferase activity, second one is alpha-1,6 glucosidase activity. In transferase activity, the enzyme removes and transfers terminal 3 of the 4 glucose residue. It transfers this moiety intake to the non-reducing end of another branch. It involves cleaving of an alpha-1,4 linkage and formation of a new alpha-1,4 linkage in another branch. This action leaves a single glucose at the alpha-1,6 branch. In alpha-1,6 glucosidase activity enzyme removes the single glucose residue which is remaining branch branch point by the alpha 1,6 glucosidase activity of the same debranching enzyme. 91% of the glycogen residue are converted to glucose 1-phosphate by the combined activity of glycogen phosphorylase and glycogen debranching enzyme. Remaining about 8% are converted to glucose by the alpha 1,6 glucosidase activity of the glycogen debranching enzyme. Now let's try to understand the third part of the glycogenolysis. Glucose 6-phosphate formation from glucose 1-phosphate by phosphoglucomutase. Glucose 1-phosphate is converted to glucose 6-phosphate by phosphoglucomutase. Active site of the active phosphoglucomutase molecule has a phosphorylated serine residue. The phosphoryl group is transferred from the amino acid serine to the hydroxyl group of a glucose 1-phosphate. It results into the formation of intermediate called glucose 1,6-bisphosphate. The phosphoryl group from the C1 carbon number 1 of a glucose 1,6-biphosphate is then transferred to the serine residue of the enzyme. It results into the formation of glucose 6-phosphate and the regeneration of the enzyme. This reaction is reversible. It allows the interconversion of glucose 6-phosphate and glucose 1-phosphate. This is very important. Phosphoglucomutase is also required for this reaction. This mainly takes place in liver, kidney and intestine. The phosphate group of a glucose 6-phosphate is removed by the enzyme glucose 6-phosphatase, and the free glucose exits the cell via the membrane localized GLUT2 glucose transporters. That is formation of glucose 6-phosphate from glucose 1-phosphate. You can see that glucose 1-phosphate is converted to glucose 1,6-bisphosphate by the enzyme called phosphoglucomutase. Over here, phosphoryl group from the serine residue is transferred to glucose 1-phosphate. Now this glucose 1,6-bisphosphate is converted to the glucose 6-phosphate with the help of again the enzyme phosphoglucomutase. Over here, phosphoryl group from the glucose 1,6-phosphate transport to serine residue of enzyme. Glucose 1-phosphate formation from a non-reducing end of a glycogen by glycogen phosphorylase. It is carried out by the phosphorylysis reaction involving the breakdown of a large molecule and forming a smaller units. Such breakdowns of the bonds by addition of orthophosphate is referred as a phosphorylysis. That is the way by which glycogen phosphorylase carry out the work. A hydrolysis reaction also involves the same process, but it uses water instead of phosphate. Glycogen phosphorylase, it's an allosteric enzyme, so obviously it has a allosteric activators like ATP, glucose 6-phosphate, glucose itself is an allosteric inhibitor. It is also regulated by covalent modification. We will discuss the same in the regulation of the glycogen degradations, that is in module 26. Removal of alpha 1,6 branches from the glycogen is carried out debranching enzymes. In glycogen, alpha 1,6 glycosidic bonds at the branch point are susceptible to cleavage by a glycogen phosphorylase, while it can act continuously until it reaches the 4-glucose molecule. Means glycogen phosphorylase stops its activity when it away from only 4 molecules from the alpha 1,6 branch point. Further degradation of glycogen is carried out with the help of debranching enzymes. Debranching enzymes has a 2 kind of activity: transferase activity, alpha 1,6 glucosidase activity. The glucose 6-phosphate formation from the glucose 1-phosphate by phosphoglucomutase, and glucose 1-phosphate is converted to glucose 6-phosphate, again that is a reversible reactions. Active sites of the active phosphoglucomutase molecule has a phosphorylated serine residue. The phosphoryl group is transferred from the amino acid serine to the hydroxyl group of a glucose 1-phosphate. It results into the formation of glucose 1-phosphate. The phosphoryl group from the C1 of the glucose 1,6-bisphosphate is then transferred to the serine residue of the enzyme. It results into the formation of glucose 6-phosphate and regeneration of the enzymes. These are the reversible reaction. It is very important. Phosphoglucomutase is also required for this reaction. It mainly takes place in the liver, kidney and intestine. The phosphate group of a glucose 6-phosphate is removed by the enzyme glucose 6-phosphatase through a Gluds transporters. Now let's try to understand, revise what we learn in today's lecture. Glycogenolysis or glycogen breakdown is a process that releases glucose whenever it is required. In liver, glycogen is stored form of a glucose for the regular maintenance of blood glucose levels. So whenever we require an energy that is available from the muscle glycogen as well as we may get it from the liver. Glycogenolysis preliminary observed during fasting, between meals or during extensive physical exercise. Obviously, the breakdown of glycogen in our body will occur as and when we require the glucose molecule. So, the glycogenolysis occurs in between the meal, during the exercise. In the skeletal muscle, glycogen is potential energy resource for muscle contraction. The glycogen present in our muscles provides sufficient energy for the movement of the muscles. So they play a crucial role in the movement of the body. The process is under stringent hormonal regulation. That regulation is stimulated by hormone glucagon and epinephrine. That is nothing but the adrenaline. You must have learned the flight reaction, flight response. At that time, adrenaline concentration in our body increases. The glucagon plays the same story. Actually, glucagon and insulin have the opposite actions. Insulin decreases the blood glucose while glucagon increases. So it is of the adrenaline that is released during the flight response of our body, which is responsible for the glycogenolysis. Certain rare but inheritable diseases of glycogen storage results in abnormal glycogenolysis. So there are certain glycogen storage diseases that might get the reference of these diseases within this paper of biochemistry. This glycogen storage disease certain are breakdown of glycogen. McArdle disease, that is a glycogen storage disease type 5, is such a disorder due to a lack of an enzyme glycogen phosphorylase that shows impaired glycogenolysis. Over here, the glycogen phosphorylase is not present. So the patient which is having a McArdle disease, in such cases, glycogenolysis is disturbed. It results into inadequate energy supply during physical activities and stress. Pori, the fourth disease, that is a glycogen storage disease type 3, is a genetic disorder arising from the point of mutation in a gene responsible for the secretion of glycogen debranching enzyme. So over here, the glycogen debranching secretion is affected. So certainly glycogenolysis is affected. It further damages the liver and muscle by accumulation of incomplete breakdown product of glycogenolysis. So students, glycogenolysis is very important for the harmony of our body. If impaired glycogenolysis is there, then we may have certain diseased condition. So students, let us now conclude what we have learned in this module. Glycogenolysis or a glycogen breakdown is one in the same, is a process that releases glucose whenever it is required. In liver, glycogen is a stored form of a glucose for the regular maintenance of blood glucose level. So whenever we require glucose, the liver will provide the glucose by the process of glycogenolysis because liver stores the glucose. Glycogenolysis preliminarily observed during the fasting between the meals or during the extensive physical exercise. Obviously, when we do a fasting, we require energy, and that energy is not available from the food, so it is provided by the glycogenolysis. Similarly, when we do exercise, we require energy, that is also available by the glycogenolysis. In the skeletal muscles, glycogen is potential energy resource for muscular contraction. Whatever the muscular contraction occurs that requires energy, and that energy comes from where? It comes from the glycogenolysis of the glycogen that is stored in the muscles. The process is under stringent hormonal regulation. Regularly it is stimulated by the hormones like glucagon and epinephrine. Certain rare but inheritable diseases of a glycogen storage results in abnormal glycogenolysis, like McArdle disease, that is a glycogen storage disease 5, is such a disorder due to lack of an enzyme glycogen phosphorylase that shows impaired glycogenolysis. You will get better idea of this glycogen storage diseases in the module 28 and 29 of the same paper. This result in inadequate energy supply during the physical activities and stress. Specifically, we are talking about the McArdle's disease. Similarly, Corey-Forbes disease, where it is also termed as glycogen storage disease type 3. It is also a genetic disorder arise due to the mutation in the gene of a debranching enzymes. It further damages the liver. So, there are glycogen defects in the glycogenolysis, and it ultimately leads to what? To diseases. So, glycogenolysis involves the two enzymes. Glycogenolysis occurs in the muscles, in the liver, but their purposes are different. That's all from my side. Thank you very much.